my cutest little children out there. Let me just check if at all I am audible as well as visible. Hello, hello, hello. Yes, I think I am perfectly audible as well as visible. And everyone, Baba, who is watching this video, make sure you send it to your friends also and ask them also to, what you say, join the live as well. Just a second, I'll put it in the WhatsApp channel and it's done. Cool. So how are you all? I hope my dearest children are doing fine. Baba, say a hi and bye and things. Do not say bye, just say hi. That will be really, really good to see all of you. So I believe that a lot of your FA1 biology examination is done. If not done, then make sure you check out the videos that are already there in the channel. I had done 2 FA marathons out there and I think it was more than enough. Apart from that, there were one-shot videos, so many things. And I really appreciate every one of you who would come and talk to me in this particular live. The reason being that only if we talk we are going to become good friends. Only if at all we are going to become good friends, we are going to enjoy the whole process. Okay, so my name is Jasmine, for those of you who do not know me, and I teach biology. And over here we are going to hello motivational. I remember you. How are you? I really hope you're doing great, and I hope your FA examination went good. Was it good? Was it bad? Was it okay? So over here in this particular session, we are going to go ahead with the chapter Principles of Inheritance and Variation. This is a series, and in a series, one chapter is basically divided into multiple parts and it is being done so that each and every concept is being super clear to you. Okay, so it is going to be a descriptive way of learning things. What I am aiming for today is basically we are going to cover Mendel's, genetics. Okay, the introduction part of the genetics. We are going to go ahead with the monohybrid cross. We are going to see what is test cross and everything. Actually, my exams are in September. Oh, is it? So you have your midterms directly, is it? That's what I'm hoping so. Anyway, chalo, let's begin. So starting off with the first thing that is there, meet Mr. Gregor Mendel. His actual name was Gregor Johann Mendel and he was actually a monk, an Austrian monk. But this monk had a special interest. He was into what finding out things. He was a curious person, which is exactly why we are gonna talk about him today, even after years. Okay, even he's no longer with us, but apparently we are talking about him today in this particular class. And obviously you might have this question in your head, that is, but why do we have to meet him? Why do we have to talk about him? Because he did what most of us do not. Can you guess what he might have done? Because he did something that most of us do not, and I'll give you the plain answer for this. He did not give up. So you know what, he was working on an experiment. And this experiment was not for few days, not for 5 days, not for 7 days, not for a month, not for an year, but actually 7 long years. Just think that you are investing your time for 7 long years for finding out something. Do you think practically it's possible for you? Do you have that much patience to go ahead and make sure that you crack that in? Well, we human beings are generally not that. Okay, we want immediate results. But he being a human being, he still did it. There were people who said that, hey Mendel, you cannot do it. There were people who said that, Mendel, you failed again. There were people who said that, how will you manage the expense and everything while you are doing all these experiments out there? But he kept on going, and as I said, for how many years? For 7 long years. One very important KCET question. That is, for how many years did he experiment things? Is basically 7 years he went on experimenting things, and the years you have to remember it. He started working on his experiment from the year 1856, and if you add plus 7, it comes out to be 1863. If you think remembering all these numbers are difficult for you, just remember at least this one. If you look at this particular number, you can see 1856. So instead of 1, if you just remove 1, forget 1, okay? 8 and 5 and 6 is there. 7 is the only one which is not there. 7 is the only one which is not there. You can remember like that. So it is 8, 5 and 6. 8, 5 and 6. 1856 he started working on an experiment and it went up to 1863. Please remember the number of years. That is what? 7 long years. Got it? Now we are going to go ahead and talk about some other thing. Basically, he did this experiment in 1863. He finished this off, but he actually got so many backlashes. There were people who said that your findings were actually not true. What I am finding is something different. It's varied. It's not exactly what you have said. And people gave their backlashes, and this particular person was not recognized for so many years. And after 37 years, just imagine that you did something right now. Okay, suppose motivational did something today, and it is a very, very great thing. It's going to change up a lot of things in our country and world and things like that. But you do not get your recognition now. Later, after 37 years, but which probably we do not know whether you're alive or not, at that particular time you are being recognized. So after 37 years later on, my dear children, 3 different people came together. Their name was Correns, Shemak, as well as Reivreis. You're going to study about them in the later part of this chapter. They said that what the Mendel guy told was actually correct. And that's when Mendel re-established his all recognitions and stuff like that. So the bottom line of the story, this particular thing, before I go ahead with the whole chapter, is basically that you do not have to give up. We have to work really hard. We have to give our best. Yes, it might not show up the results in 1 day or 2 days or probably a week. It might take time, but it is going to definitely show up. Yeah, and finally in your life you're going to have ups and downs, but always remember, if you have done something good, okay, if you have done something good, if you have put in your hard work, one day or the other, it is going to be recognized, as you saw in Mendel's story. Now, over here, the question is, finally, Gregor Mendel, what did he become? He became the father of modern genetics. Please remember, who is the father of modern genetics? The answer is pretty simple. It is what? Gregor Mendel. His actual name is Gregor Johann Mendel. Okay, his actual name is Gregor Johann Mendel. Okay, now the question that I have to you guys, and you have to answer me, ma'am, my name is Priya. Okay, Priya, I'll try to remember your name like Priya itself, even though I see motivational. You are actually having a very motivating name out there. Anyways, Priya, so my question to you, since we have studied a little part of this particular chapter, from which year to which year did Gregor Mendel do his experiment? Any guesses? From which year to which year did Gregor Mendel do his experiment? And by the way, everyone who is watching this video can also answer this, because the reason I'm asking you this question is, it's a KCET question. Okay, it can be coming in your KCET, it can be coming in your second PUC, MCQ are there for 7 years. Okay, 7 years, perfect, Priya. And 7 years started from which year? From which year did he start his journey? From which year did he start his journey? And I hope you do not mind asking questions like this, right? Children, it's just to engage you guys. And remember, even if you give me wrong answers, I'm not going to do anything to you. I'm Papa person da. So 7 years, starting from 1856 to 1860. Perfecto. Awesome. Super good. Now we go ahead with the basics of genetics. Okay, the basic part of genetics that we all should know. If we do not know it, this basically we cannot go ahead with the whole chapter. So the basics of genetics goes like this. I'm going to make up something over here. Listen, for everything in our body, we say it's a character. For example, height is there. Okay, height is what? Height is basically a character. Now, each character which is there, it can be having different types of it. For example, height, there is height which is tall, and there is height which is dwarf. Okay, height which is tall and height which is dwarf. So basically, these are 2 types of height, 2 types of one particular character. I call these things as traits. Okay, the types of the one particular character, they are known as traits. For example, I have what? Tall and dwarf. Tall and dwarf. Okay, now who decides that basically what is going to be my character? Who decides what is going to be Priya's character, as in Priya's height, Priya's skin color, Priya's eye color, or Jasmine Ma'am's skin color, eye color, height, and things like that? Basically, our genes are going to decide that. Ma'am, what do you mean by genes? I do not understand this genes and things like that. Dominant, yes, we're coming to that. So listen to me very carefully. We all have chromosomes. Okay, you have chromosomes, I have chromosomes, we all have chromosomes. How many chromosomes do we have in human beings? So I'm going to make a thing like this over here, and I'm going to talk about human beings over here. How many chromosomes do we have, Baba? Any idea? How many chromosomes do we have? We have almost 46 number of chromosomes. Okay, we have 46 number of chromosomes, and these 46 number of chromosomes, they come in pair. Okay, they are not alone 1 1 1 1 1. They're going to come in pair. So basically, I'll say that they are 23 pairs. How many pair do we have? We have 23 pair. And each chromosome, suppose this is one chromosome, this is going to be another chromosome. I'll say this is what? One pair. In one pair of chromosome, how many chromosomes are there? 1 and 2. So in that way, I'm saying in 23 pairs, in 23 pair, total, if at all you count 111 chromosomes are there are 46 number of chromosomes. Yes, yes. Okay, ma'am. And Priya also said 23 in pairs. Perfect. Now, I'm going to deep dive into these chromosomes and I'm going to talk about one very important thing. When you see the chromosomes, what are chromosomes? This is nothing but your DNA. This is your DNA which is going to be compactly arranged. Your DNA is compactly arranged and forming a structure like this, which you actually call as chromosomes. If at all, I take this DNA and make it go like this. Okay, I take this DNA and I'm like, okay, let me stretch it up and see what's in there. It is going to be looking like this. And each part of our DNA is basically having some codes. Just like you have microchips and all in your, not in your body, in your mobile phone and stuff like that, no? Similarly, this is a microchip that we have. The DNA is a microchip that we have. It stores all the information. How? I'll tell you. So suppose this is a segment of DNA that I have. This is another segment of DNA. This is another segment of DNA. Each segments over here will be storing certain information to form what? To form protein. So this can be forming protein number 1, this can be forming protein number 2, this can be forming protein number 3. And these 3 different proteins might be responsible for different characters. Probably this protein is super important for deciding the height. This protein could be super important for deciding my eye color. This protein could be super important for deciding my skin color. Right? So each segments of this particular DNA, what do I call it? I actually have a name for it. I call this as gene. So this is a gene, this is another gene, this is another gene. So we have segments, okay, we have segments of our DNA. We call them as genes, and genes are responsible for what? For the production of proteins, for coding of proteins. And these proteins are going to have a direct or indirect effect on our height. Probably this protein is super important for someone for production of a growth hormone. This protein is super important for production of a growth hormone. This protein, when this protein is there, it helps in conversion of A to B, and this can help in giving me height. Okay, when the growth hormone is formed, basically I get too much amount of height. That's the basic logic. So all of them are responsible for each of the characters. Now I'm telling you one very important thing over here, that when I say a pair of chromosomes, one chromosome I actually get from my father. Okay, another chromosome that I have, I actually get from my mother. So there is father and mother who is contributing there is one that is coming from either of my parents. Okay, and basically when it comes, you need to understand that if in this particular position I have a gene coding for height, similarly here also I would be having a gene coding for my height. I would be having gene at the same location on both of these chromosomes coding for my height, right? And the proteins over here can be different. Probably this gene over here will be producing protein 1 in too much amount. Okay, it has the information to produce the protein 1 in too much amount. Whereas this particular gene that I have for the height itself, this is also producing protein 1 only, but this protein 1 which is produced is in too less amount. So these two varieties of a gene, what do I call it? Have you understood it? These are two varieties of what? Of one thing, that is a gene that is coding for height. Similarly, as I said, one character is there, you have 2 traits, right? You have 2 traits. Similarly, one gene is there, gene is for height. This is a gene for height, this is a gene for height. But both of them are coding differently. One codes for more protein, one codes for less protein. So what do you call different type of genes are there? For one particular character, you have different type of genes. What do you call it? You have a term for it. Any guesses? Sakiba ma'am, I have one doubt related to counseling process. Sakiba, I'll request you, even though I love to help you, I'll request you if you can put down a comment in the comment section, or probably just wait for some time. Okay, if you can come back at 5:30 and ask me, it will be really, or probably you can just put down the question and come back at 5:30 and I'll give you the answer. Will that work? So 5:30 I'm planning for a choo-choo break. Guys, I'm asking you a question. Any idea? Over here, you have two genes. Okay, both of them are coding for your height itself, but there are two varieties of thing. One gene is coding for more proteins, one gene is coding for lesser protein. What do I call these two different type of genes? Pawan, very good evening, dear. What do I call these two different type of genes? Come on, Chutku people. Fast. I have a term for it. It starts with A. It starts with A. Now can you guess it? Two different varieties of a single gene is known as what? It starts with A. I have given you the clue. If at all you do not know, you tell. Ma'am, you only tell. Abnormality. Ayyo. Okay, it is known as allele. It is known as what? Allele. Well, it's okay. It is going to be known as what? Allele. Okay, so over here there is a gene. Both of them are here itself. These genes are for height only. Similarly, Baba, here also you will be having genes. These genes could be for different things. So I'll say these two are alleles for skin color. Okay, similarly I have genes over here. I'll just change the color such that it does not cause any confusion. Okay, there is a gene over here also. This is for my skin color. This is for eye color, this is for my skin color. Both of these are genes, but they are varieties of genes that we have. What do you call them as? They are alleles for skin color. These are alleles for eye color. These are alleles for what? Your height out there. Similarly, for each of the gene, you would be having varieties. You call them as alleles. Have you got it? Understood this? Understood? If yes, can you give me a thumbs up? If no, can you ask me the doubt? Yes, ma'am. Perfect. Thank you, sir. Perfect. That is a logic that we have to apply over here. Okay, so this has been done. I spoke about few things. That character is basically a broader term. That means height, or it can be anything. Traits are basically types of character. So under height you have tall and dwarf. These are the traits. These are going to be known as traits. And basically each of these traits would be having alleles. Each of these traits would be coded by alleles. Okay, so a gene, the different type of genes that you have, what are they known as? They are known as alleles. Now I'm going to talk about one more very important thing. Listen to me very carefully. When I talk about alleles, this particular allele which was there, okay, this particular type of gene which was there, it was able to produce protein 1 in way too much amount. Okay, so I'll say that basically it is capital T allele. This particular allele is there which is producing protein 1 but in way too less amount. So what do I call it? I call it as small t. Just for denoting it, I'll say it's capital T gene and it is going to be small t gene. Okay, over here, who is going to express itself? Which one will be able to express itself? Ma'am, both of them are expressing itself because this is coding for more protein, this is coding for lesser protein. But anyway, it's not like but the protein which was being produced, it is lessened up. It's basically it's producing a little amount of protein. But both of them are producing something, right? Which one will show more of its effect? Obviously this one, because it is producing more protein, it will be showing its effect. So I'll say there are alleles whenever you have dissimilar pair of alleles, whenever you have different types of alleles for one particular character, one allele, exactly Pawan, one allele which is able to express itself is going to be known as what, Baba? They are going to be known as dominant allele. They are going to be known as dominant allele. And the one which is not able to express itself, they can be producing protein, they may not be producing protein at all. The one which is not able to express itself fully, what do you call this as? Come on, you only tell me. The other one which is not able to express much, what do you call this as? Any idea? Just give me a second. Recessive allele, exactly. So over here, what do I call this as? I will call this as recessive allele. Got it? So we have dominant allele and we have recessive allele. Tada. So these are the basic things which are there. Now probably I can go up and talk more about all the things. These are the basic things that you needed to know. Always remember, I get one chromosome from one of my parent, another chromosome from another parent. That is how basically I get 46 number of chromosomes. So when we say 23 pair, okay, 23 pair means 46 number of chromosomes. 23 are there in the sperms. 23 chromosomes are there in the egg. When both of them fuse, that's when they become a pair. That's when they become pair, and that's how we get 23 pair. Okay, and each chromosome comes from each of my parents. Not forget it. It's very, very, very important. Cool, cool. Now let's go ahead and talk about the next thing out there. Just a second. Where am I? Okay, let's talk about things in written format such that things become even more clear. You look tired, I look tired because probably I have tied my hair. Nothing else, Macha, just a second, okay, it's too hot over here. It's not working, Come on. Work. Okay, apparently it will work slowly. Anyways. Cool. So over here, what do we have? Mendel proposed that traits were specified by heritable elements called factors. What? So basically Mendel said that the traits or the character that we have, it is specified by the heritable elements. That means something that we can inherit from our parents, and you call them as factors. Actually, they were not called as factors. They were known as genes. But Mendel didn't know it. Okay. You actually call them as gene right now. Back then, since Mendel Papa guy, he was working without microscope and things like that, he called this as factor. Okay, done, ma'am. Factors come in different versions or alleles, or we can say that alternate forms of a factor is known as alleles. That means genes which are there, they come in pairs, different versions. As I said, some can produce more amount of proteins, some can produce less amount of proteins, some can produce defective proteins. Basically, I'll call all these varieties of a gene as what? Alleles. That is also done, ma'am. Okay. Alleles are of two types: dominant alleles and recessive alleles. Okay. Dominant being expressed over the recessive allele. Recessive alleles are only expressed when no dominant allele is present. So, to come to talk about this, I'll need some time. Probably you will understand things more clearly over here. So over here, just imagine scenario number 1. Scenario number 1 goes like this. By the way, everything is clear until this point? If it is, can you give me a thumbs up? If my children, everything is clear until this particular point, can you quickly give me a thumbs up? Yes, ma'am. Understood. Perfect. Cool. So scenario number 1 goes like this, where what do you have? You have one chromosome in the egg comes from the female. This is the chromosome that is coming from the female. It is having capital T allele. Okay, probably it is going to produce too much amount of proteins out there. Okay, another chromosome in the sperm comes from the male. Okay, and here the allele is what? Small t. When both of them comes together, I get a combination that is capital T, small t. Capital T, small t, this is the combination. Okay, now I'm going to talk about two very important terms that you need to remember. One is known as genotype, and the other one is known as phenotype. Okay, there is genotype and there is phenotype. What do you mean by genotype over here? Genotype means their genetic makeup. Can you see my genetic makeup? Can anyone see my genetic makeup? I cannot see Pawan's genetic makeup, nor can I see Priya's genetic makeup. I cannot see anyone's genetic makeup. Why? Because it is there inside the cell. Inside the cell, Baba, there is the chromosomes, and the chromosomes have DNA. The DNA has all the information are there. I cannot see all of it, right? I cannot see it. So this is genetic makeup. Exactly, Pawan. So over here, what is the genetic makeup of this particular person? What is the genetic makeup of this particular person? If at all you have been asked, there is capital T and there is small t. What is the genotype that is going to be there? Pawan told me what's phenotype also. Perfect. Da. What is the genotype of this particular person? Any guesses? I have capital T and small t. So what will I say is the genotype? What will I say is the genotype? Come on, I'm waiting. He or she got one from heterozygous start. Okay. Oh my God, heterozygous. We'll come to that. Okay. So anyways, genotype, if at all be asked, genotype means genetic makeup. I will say it is capital T, small t. I'll say it is what? Capital T, small t. Now if I'm asking you, what is a phenotype? What is a phenotype? Phenotype means physical appearance. This is what I can see. Oh, Pawan is this tall. Oh, Pawan is this short. Or Jasmine Ma'am is this tall. Or Jasmine Ma'am is this short. I can see this. What is the physical nature over here? What is the physical appearance that I am going to see? So P for physical nature, P for physical appearance, you can remember like that. Over here, Baba, since you have one capital T and there is one small t, and I told you capital T means basically it is able to produce too much amount of proteins. It is a gene that is producing too much amount of proteins. It will be able to express itself, which is why basically we are going to be tall. It is going to be dominant over the small t, and I'll say the phenotype that we are going to have is what? Tall. So genotype is capital T small t and the phenotype is going to be what? Tall. And I am coming to heterozygous part also. Just wait for a second. I hope this part is clear. Okay, now I go to scenario 2. Scenario 2, which is there, it is like this. From the female you get capital T, from the male also you get capital T. From the female and from the male you get capital T. Over here you have got a pair of chromosome. Now here I am asking you something. Please be very careful and give me the answer. What is the genotype? What is a genotype? I'm not talking heterozygous, homozygous, things like that. I'm just asking you what is the genotype of this particular person out there. There is capital T and there is capital T. What is the genotype of this particular person? Come on, Pawan. Try Madi. Any idea? And anyone who probably is watching this video, Baba, you can also try. Capital T and capital T. Exactly. It is going to be what? Capital T and capital T. That is a genetic makeup. Can I see it, ma'am? No, ma'am. Obviously, no. Okay, guys, I'm asking you another question. What is a phenotype? What is the physical appearance of a person who has both capital T and capital T? What is going to be the phenotype, physical appearance? And anyone can answer. Pawan, by the way, you got that correct. Okay. Tall is the phenotype. Exactly. Exactly. It is going to be tall. It is going to be tall, right? So that is actually perfect. That is actually perfect. So over here, genotype is capital T, phenotype is tall. So ma'am, I have a doubt over here. Previously, you said this is also tall, right? In this condition where the genotype was like this, you are saying this is also tall. How do we, I mean, like, I do not know what do you call the technical term as. So listen to me very carefully. As Pawan told, whenever you have two sets of alleles and they are different, you do not have the similar ones, you have different ones. One is capital T, another one is small t. The phenotype is tall. They are appearing to be tall. Why? Because this is dominant over the recessive one. This is dominant over the another allele. You call this condition as heterozygous condition. Heterozygous condition. Heterozygous means both the alleles are going to be dissimilar. They are going to be dissimilar. Okay, cool? Yes. Now talking about over here, what do you have? Here, again, when you see there is capital T, and it is also tall, you have a term for it. When both the alleles are same, you call this as homozygous. Okay, you are going to call it homozygous. Homozygous means same. Get it? So there could be heterozygous tall, as Pawan told before, which looks like this, or there could be homozygous tall, which could be looking like this. Cool? Yes? Works? Perfect? Yes. Now what's the charcha over here? Teaching 9 students before. No, it's not about numbers, it's about quality of teaching. See, oh my God. Thank you so much, Pawan. That's so sweet of you. Even I did not have to go ahead and answer to that question out there. So listen to me. It's not about the number of children who is, as he said correctly, it's not about the number of children who are watching this video. It's about who is watching it with their whole heart and who wants to learn. Even if it's 1 person or probably 0 person, I'll still go ahead and teach. Why? Because I'm a teacher. And a teacher has to make sure that whoever watches this particular video later on understand it in the best of their capacity. You get it? So thank you, Pawan, for answering from my side. It's so sweet of you. And peaceful guy, Baba. Guess we have to be motivated. We cannot be sad about things, right? Life is very short to be sad about things. Come on, Macha, you also enjoy. Okay. Now we're coming to the third scenario, and that is this. So from one parent, you're getting small t. From another parent, you're getting small t. And both the parents brings up something like this. Now I have a question to you, Baba. What is going to be the genotype in this particular case? What is going to be the genotype in this particular case? Come on, everyone, try Macha. Okay, genotype is small t. Fine. What's the phenotype? Genotype is absolutely correct, Pawan. What's the phenotype? Physical appearance? And Baba, everyone who is watching this video have to, Peaceful says it is small. Exactly, it is going to be dwarf in nature. That's the spirit. So over here, I'll say genotype which is there, it is going to be small t, and phenotype which is there, it is going to be what? It is going to be dwarf. You call this term as dwarf. Okay, that is a scientific term for it. Now over here, I have a question to all of you. Okay, both of you basically, and that is, here, is it heterozygous or is it homozygous? This condition is heterozygous or homozygous? What do you think? Homozygous, ma'am. Exactly. It is going to be homozygous. Why? Both the alleles are similar. Now I have one more question to ask you, and that is, do you ever find heterozygous dwarf organisms? Can you ever find heterozygous dwarf organisms? So this is homozygous dwarf. In the tall, no, there is heterozygous tall and homozygous tall. Here, can you find it? Yes, exactly. Peaceful as well as Pavan. No, ma'am, you cannot see that. Why? Because if I say it's heterozygous, that means there will be someone else who is dominant and it will express over itself, which is why it cannot be remaining dwarf. So this is a basic funda and you have understood it very, very clearly. So always remember, the recessive characters are going to show up only when they are homozygous. The dominant characters which are there, they are going to be showing up even if there is heterozygous, or it is going to be, what do you say, homozygous out there. Until this particular point, is it super clear to you? If yes, can you quickly give me a thumbs up? If yes, can you quickly give me a thumbs up? And you children are awesome, okay? Yes, ma'am. Understood. Perfect. Thank you so much. Peaceful. I hope you are also understanding, Macha. Okay. Now we go ahead with the next thing. Where is the next thing? Ayo. Yes. So over here, what did we study about? In most sexually reproducing organism, each individual has two alleles for a gene. I told you, one gene from one parent, one gene from one parent. This pair of alleles, the genetic representation is known as genotype, and it determines the physical appearance, which you call as what? The phenotype. That is also clear. We studied about all this thing, right? By the way, everyone who wants note, make sure you look out for the AIO page. AIO page is there. I'm going to talk about that very shortly. Just listen to me. Now there are two types of cross. There is monohybrid cross and there is dihybrid cross in your NCERT. Okay, I'll tell you what it means. Monohybrid cross means, Baba, I am talking about only one character, that is height. Okay, I'm talking about one character, a person who has tall height. I'm going to mate it with another person who is having dwarf height. And I'm going to see what are the hybrids, what are the new things that are going to be formed over here. This particular cross that I have, considering only one character at a time, is known as monohybrid cross. Okay. Dihybrid means I am not considering one character, I am considering two character. For example, I am talking about how is going to be the inheritance if I talk about height, okay, and also the shape of the seed. The shape of the seed and the height together, I am going to see how this is going to be inherited. So two characters if I study, it is going to be a dihybrid cross experiment. Why do you call it dihybrid? Because in the first one, okay, you have one particular character you're fusing, but now when they fuse, you might get new characters, you might get new combination. That is why you call it hybrid. Okay, so dihybrid is considering two characters at the same time. Monohybrid is considering only one character at one particular time. Krishna, hello dear, and I hope you're doing good. And by the way, every one of you is watching this video and understanding things well, make sure you hit the subscribe button. In this particular video, we are going to talk about what? The dihybrid experiment. Okay, sorry, not dihybrid, it's basically monohybrid experiment. And before we go into that, there is one very important thing, that is basically the garden pea. So Mendel, who was working really hard, he was working on a plant which you call as garden pea, but there is a scientific name for it which you have to remember. Anyone knows the scientific name? Krishnaholic, Pavan, Peaceful, or probably Priya? Anyone knows the scientific name for this? Of garden pea? Anyone can guess, by the way. Pisum sativa. Who said that? Perfect. It's going to be known as pisum. Ayo, I was writing pistum. So it is known as pisum sativa, and we have to make sure that the second letter is going to be small. Okay, why? Because it's a specific name out there. So genetic name, genus name, and the specific name. Anyways, so we have this particular plant, and in this particular plant, why did Mendel choose this particular plant? Because he said, okay, Pisum sativa. Everyone said. Krishna also. Perfect. Why did he choose Pisum sativa? Any idea? I'll tell you. Okay, you can also keep on guessing. I'll tell you few points. First thing, basically it has a very short life cycle. So if at all I want to fuse them and I want to see how their babies are going to look like, I do not have to wait for years. I can clearly see that. Okay, I can clearly so fast see that. Peaceful says they have contrasting traits. Exactly. That's the next point. Basically, this particular plant has so many characters to study. and all the characters which are there, they are having contrasting traits. As I told you, if height is there, height is having two different types, tall and dwarf. They are very, very contrasting. I can clearly see of a dwarf plant or a tall plant is being born. Similarly, you have several other things that is there. Okay, so you have shorter lifespan, and basically, as I said, Peaceful said, they have contrasting traits. Any other things? Any other things that you have an example as? They are basically going to have different type of flowers, which you call as bisexual flowers. Basically, they have the male and the female part in the same flower. So you do not have to do pollination. They are going to do self-pollinate. Okay, they are going to do self-pollination very, very easily. So you do not have to be worried about them pollinating, fertilizing and producing progenies out there. Okay, cool, works. Yes, so these are few points which are there. Anyway, the contrasting trait part is the most important thing that we have to study over here. So over here, how many characters were there? Any guesses? In Pisum sativa, or basically garden pea, how many contrasting characters were present? Or basically, yeah, characters were present. We have a number, 7, exactly. So there are 7 pair of contrasting characters. When I say 7 pair, okay, when I say 7 pair, you have to be very, what do you say? We need to learn the chromosome numbers for those traits, ma'am. Pawan, no, Baba, you do not have to learn. Okay, 7, ma'am. Okay, so Pawan is also saying 7, and Krishnaholic also saying 7. Yash is saying 14. Yes, this is what I wanted. Yash, listen to me very carefully. So 7 pairs of character is there. 7 pair of character is there, but in each character you have 2 traits. In each character, Baba, you have 2 traits. Means, as I told you, height has 2 traits. Similarly, each character which you want to see are going to have 2 traits. That is why you say 7 into 2, basically 14 traits are there. So 7 pair of characters and 14 traits. Always remember, this could be a KCET question and you can mess it around. Okay, so I hope this is clear. Yeah, we're going to see one by one so that it becomes all the more clear to us. The most easy one to remember is going to be the stem height. This is what a character. Okay, this is a character and I'm going to write the trait over here. This is trait 1 and this is trait 2. Okay, in stem height, what are the 2 traits that you have, Baba? It's very, very simple. Tall and dwarf. Exactly. There is going to be tall and there is going to be dwarf. Done, ma'am. Okay, done. That is one thing. Now we are going to the next thing, that is basically flowers. Okay, so I have flower color. Flower color, that is the first thing that I have. In flower color, how many flower colors are there? How many flower colors are going to be there? Violet or white? Violet or white? Pawan says violet or white. Peaceful says, I cried KCET with 6,600. Oh my God, that's superb. Okay, Yash also says 2. So listen to me. There is violet flowers and there is going to be white flowers. Cool. Here, flower has one more character that we study. That is flower position. That is going to be flower position. Any idea what are the 2 different type of flower positions that we have? What are the flower positions that we have? Axial and terminal. Exactly. So we have 2 positions. One is axial and one is going to be terminal. For those of you who are wondering what is axial and what is terminal, listen to me very, very carefully. Okay. So over here, I have this. Okay, I have this. Suppose this is a stem that we have, and from this point I get a branch. But at this particular position, I have a flower. This particular position, I have a flower. This is known as axial flower. Similarly, if at all I am getting a flower at the tip of something, I'll call this as terminal flower. So we have 2 type of flower positions. One is axial position and one is going to be terminal positions. Okay, axillary and terminal, that is the other word that we have. Yes, Priya, you are also correct. And by the way, backbencher, I could not decode your entire thing, so sorry. Anyway, so we have 2 things for flower. Now we go ahead with the next thing, that is seed. In seed, Baba, you have shape. In seed, you have shape. You have 2 type of shapes. Any idea? What are the 2 shapes that we have in seed? What are the 2 shapes that we have in seed? Guess, guess, guess. Round and wrinkled. Round and wrinkled. So I'll just write round, and over here It is going to be wrinkled. Perfect. What do you mean by that? Basically, Baba, the seed can be like this or they can be like this as well. Okay, they can be papa guys, they are not drinking water enough, right? So they can be wrinkled or they can be round. Now in seed, one more thing is there, seed color. And this is something that is so confusing for a lot of you guys. In seed color, Baba, you have two things. And by the way, Priya, motivational, Krishnaholic, all of you are doing absolutely good, and also my peaceful. Okay, so yellow and green. Krishnaholic says yellow and the green things are there. Okay, and she wrote, I do not know whether Krishnaholic is a female or a male over here, but anyway, yellow and green, everyone says yellow first. Okay, in that way I'll write yellow and green over here. Yellow and green. And we will decode it later on, okay? But anyways, for seed also we have two characters out there. Now we go ahead with pod. What do you mean by pod? Pod means basically green piece which is there, comes on inside a sack like thing, no? Inside that you have pea. So this is the pod which is there. So in pod also we have pod color. Sorry, we'll start with pod shape first. We'll start with pod shape. In pod shape you have two things, traits. Come on. He wanted to say he can call you mine according to his long paragraph. Yes. Oh my God. Is it? Okay. So there was a, okay, that's personal information anyways. Okay, so motivational says inflated and constricted. Pavan says full and inflated. Pavan macha. Okay, full and inflated. In Krishnaholic says inflated and constricted. Listen to me very carefully. Okay, so inflated or full means the same thing. Inflated means like this only. It's completely not dry and all. It's completely like this only. This is known as inflated. It is also known as full. Inflated or full is nothing but the same thing. Inflated, full, and over here the other one that we have is constricted. Over here the other one that we have is what? Constricted. So it is going to look like this. It is going to look like this. This is the constricted version. So that I'm putting over here. Now we have one more thing right over here, and that is pod color. And that is pod color. In pod color, Baba, you have two colors. Come on. In pod color, there is one more thing that you can go ahead. This is the last one. This is the last character which is there. Green and yellow. Exactly. There is going to be green and yellow. Green and yellow. These are the traits and everything out there. And now we are going to go one by one about that. Before that, Balu, is there any update? No, dear. There is no update as of now. When it comes, I'll definitely let you know. Okay, Yash, you want to tell something? Yes. So right now, who was this particular person? Backbencher said the mine wala thing. No, so I remember. My name is Jasmine. Okay. So in Jasmine, M-I-N-E comes at the end. So one guy, when I was in which grade, I do not remember. I think 11th or 12th. I do not remember. Anyways, and that particular time, he said he didn't used to call me Jasmine. He used to call me Jasmine. So yes, so apparently he had a crush on stuff like that. But anyway, that memory came in. So over here, this is what I want to tell you. Now listen to me very, very carefully. Okay, this is very, very important. Listen to me carefully. You can get questions on what are the dominant traits and what are the recessive traits. As in, they can give you certain traits out there. For example, round seed shape and constricted pod shape and things like that. So many, 4 options can be given, and in KCET, in MCQ, they can ask you which one is going to be a recessive trait. So you should know amongst them which are recessive and which are dominant. So over here, everything that I've shown over here, they are going to be what? They are going to be dominant one. And all of them that I've shown over here, they are going to be recessive one. And it's not like you have to muck things up. I'm going to tell you in what way you can remember things very, very easily. In stem height, tall and dwarf. Baba, tall people we can clearly see, so that becomes a dominant one. That becomes a dominant one. In flower color, violet and white. Which is the darker color? Violet one. That can be clearly spotted. So again, violet is going to be a dominant trait. Flower position, axial and terminal. This can create confusion. So listen to me. A comes before T in the alphabetic order. So A is going to be the dominant one, right? Now over here I am going to seed shape, round and wrinkled. Will you prefer eating wrinkled one or will you prefer eating round ones? Round ones, no. So we will consider that is going to be dominant. Okay, seed color, I'm going to keep it like that for some time. Pod shape, again pod shape, will you choose this when you go to market or this? Obviously this is something that you're going to choose, so I'll say inflated and full is going to be dominant out there. Now pod color, again when you see the pod, it is green in color, I will pick the green one. Will I pick the yellow one? No, I will never pick the yellow one. I'm going to pick the green ones out there. So again green is going to be dominant out there. Now listen to me, seed color is left, and usually the green peas that you eat, you call it as green because they are green in color, right? But actually there's an exception. Yellow color is going to be the dominant one, and green one which are there, they are going to be the recessive one. This is the only exception which is there, and it is super important. Motivational cells, gravity is a thing. Thank you so much. I was trying to remember what was the mnemonic for it, so it is actually gravity, and thank you so much for reminding me. So gravity, G stands for green, R stands for round, okay, A stands for axial, V stands for violet, I stands for inflated, T stands for tall, and Y stands for yellow. Okay, so yellow is going to be dominant in the seed color, always remember that. So all of you, I want you to imagine one thing right now. Okay, close your eyes, everyone close your eyes. Okay, and after closing, I was going to tell, after closing your eyes, give me thumbs up, Papa. Good. Yo. So listen to me, everyone close your eyes and just imagine green peas, okay, green pod, and inside that yellow peas are there. Yellow green peas are there. Imagined? Always imagine this while writing the answers also. Cool? That's all. Okay, that's the only way I can make you remember things out there. And yes, gravity is there, but some people will follow yellow is the dominant in this or things like that. Now you'll not forget. Cool? Works? Have you remembered everything? So these are 7 pair of character. Yesh, okay, Yesh was saying 14 out there. So these are the 7 pair of character, and these are the 14 traits that you have. So trait and character is different, always remember. So Pavan gave me thumbs up. Everyone else didn't understand it, Macha. No thumbs up for Jasmine Ma'am. Please, give me one thumbs up to make me understand that, ma'am. All cool until this particular point. Everything is super clear to me. Sachin, ma'am, I'm a dropper in application showing claim verified 0 remarks. Previous year verified 12. Okay. Class A student, what should I do? Sachin, please go ahead and try to download the verification slip. You should mostly be able to download this slip. Okay? Okay. Thank you, Yash, and motivational everyone. Thank you. Let's go ahead. Krishnaholic, clear na macha? I hope it's clear. Okay, now we are going ahead. By the way, you will not get this particular page because this is a new thing. If you want, you can take a screenshot of this. Okay? Yes. Perfect. So, I go over here. So now we come back to 1856 to 1863 story. 7 years, I told you in the starting part of this particular chapter. You can remember this year as 8567 is the only thing that is not there. So that is why 7 years. That is why 7 years. 1856, which is there. From that 7 years he worked, and until 1863 he worked. Now, there was a newspaper article. Actually, I made this particular edit. Actually, there was no newspaper article, but there was so many news that Mendel did something wrong. Okay, whatever findings he did, he was doing something wrong, and we are going to see what is that something wrong. Okay, we are going to soon see what is something wrong. So according to him, whenever a dominant trait and a recessive trait comes together, you show the dominant trait, right? So red flower and white flower, when they cross it, and suppose red is a dominant trait out there, then everyone should be red in color. All the progeny, small plants which are there, they should be red in color. But apparently things were not the same over here. Okay, some person said that that's not the case, Mr. Mendel. That's not the case. When I am crossing red and white, I am getting something else, that is pink. Okay, so over here the pink color is not visible, but actually it is pink in color. So red and white, when he crossed, he got pink. But according to Mendel, it should have been red. So now in this particular session, after going ahead with monohybrid cross, we are going to see the 2 different scenarios. Was Mendel right? Was Mendel wrong? And things like that. Cool? Okay. We're going to talk about the monohybrid cross experiment. So first of all, before going ahead with that, why did people start believing his work? This can be a question. Yes, incomplete dominance, Pavan. Yes, you're absolutely correct. So why did people start believing his work, Mendel's work? First thing, he was the first guy to use math in biology. Tomorrow I also come and say that basically 256 students studied from me and they had got this much rank, this much rank, this much rank. You're going to believe that, oh, Jasmine Ma'am makes sense. She's a very good teachers who produce so many rank holders and stuff like that. Whenever we use math in anything and analytics, we show that it becomes more believable, right? So similarly, he was the first guy to use math in biology. But I don't think I need to produce numbers out there for you to believe me. I think my teaching would be more than enough to believe, right? So first use of math in biology was done. Second thing, large sample size. So he was not like, I will cross only these plants and I'll see that what is the result. That was not his case. Basically, he used one plant, then another plant to do his experiment, then another plant. So many plants he kept on doing his experiment, which is why it was largely seen that, okay, in this plant also things were working. In this plant also, when I did the same experiment, same result is coming. So that is why large sample size was helping him in make people believe that, yes, this is actually correct. Next thing, repeated experiments. He didn't say that I'm going to go do 2 experiments and basically with that I'm going to give, conclusion. Baba, he worked for 7 long years, and for repeatedly, if you're getting the same results out there, people had to believe him. Finally, clear and contrasting traits. He chose a plant which was having clear and contrasting traits, which could again make people see and believe that there is no other scenario. It was very easy for the people to believe when they saw that he was choosing a plant, or he played with a plant that has contrasting traits. So these were the few things or reasons why Mendel's work was being believed. Now this can be a question in your second PUC. Mostly they have not asked until this particular point, but as I said, it can be. We cannot, ignore that. Now, I spoke about all of it already, but I can still talk about it. Ever wondered how passing of genes work? As I told you, we all, okay, in my body also there are 23 pairs of chromosome. Pawan also has 23 pair of chromosome. Priya also has 23 pair of chromosome. Yash also has it. Krishna also has it. Everyone have 23 pair of chromosome. Now here I am just talking about 1 pair of chromosome that this particular plant is having. In both the pair of chromosome that is there, in a pair of chromosome that is there, on this particular area I have a gene coding for height, in this particular area I have a gene coding for height. Both of the alleles are similar over here. It can be dissimilar also. It can be different also. That's not an issue. I am talking about a scenario where both of them are same. Similarly, in this particular case, again, this is suppose someone else. You have a pair of chromosome. Here also the alleles are same. As I said, this condition is known as homozygous condition. This is homozygous dwarf, and over here this is going to be homozygous tall. Anyways, from the pair of chromosome, only 1 chromosome is going to get inside a gamete. From this pair, only 1 can go to one particular gamete out there. Now here, whatever it does, it is going to be capital T only, because it just have that to give, right? Which is why it is always going to be capital T. From here also, you get always only one thing, that is going to be small t. And when you get both these chromosomes, suppose this is in the egg, this is going to be in the sperm, this is going to be in the sperm. When both of them fuse, you are going to get a pair of chromosome again, but this time the pair looks different. What do you have? This chromosome is having capital T allele and this chromosome is having small t allele. This was the basic that we already discussed, right? But I realized that lots of you had joined later on. Now over here, we are going to talk about, with this logic, we are going to talk about Mendel's experiment. So Mendel did a monohybrid cross experiment. Monohybrid means one character was being considered over here, and that was height. According to him, he had crossed 2 parents. So I'm just writing over here parents. Okay, he crossed tall parent with a dwarf parent. And always remember, both of them were homozygous. They were homozygous. Homozygous means alleles are going to be same. Homozygous. By the way, do you know the other term that you can use for homozygous? Any one of you, do you know the other term that we can use for homozygous? It starts with P. Anyone. Homozygous is also known as what? Anyone can guess? Pure. Exactly. You call this as pure breed. Okay. You can also call this as pure breed. So pure breed tall and pure breed dwarf. Anyways. So in this case, the parents were capital T since it is homozygous tall, and it is going to be small t since it is homozygous dwarf. You can also call this as pure breeding. Anyway, now we are going to form the gametes. The gametes, whatever you do, Baba, here the gamete can have capital T only. Here also it will be having only capital T, but only 1 chromosome. Similarly, it will be having small t, and here also it is going to have small t only. Nothing else could be there. Okay, ma'am. Now we are going to cross them, and we are going to get the first generation. You call the first generation as what? You also call this as filial generation. Filial 1 generation. Okay, filial 1 generation. Now we are going to cross them, and we are going to get the first generation. You call the first generation as what? You also call this as filial generation. Filial 1 generation. Okay, this is going to be what? The first generation out there. Now I'm going to cross, and you have to imagine. A sperm carrying capital T fuses with another egg carrying small t. So in this, the male gamete carrying capital T fuses with a female gamete having small t. What do you get? You get capital T, small t. Okay, ma'am. This fuses with this. Again, what do you get? Capital T. Where is it? Just a second. Okay, this has some issues. Okay. Again, what do you get? You get capital T, small t. When this fuses with this, again, what do you get? Capital T, small t. When this fuses with this, you get what? Capital T, small t. Now, I have a question for all of you. What is going to be the genotype of F1 generation? What is going to be the genotype of F1 generation out there? Any guesses? What is the genotype of F1 generation? One is to 2:1 now. Dun, dun, dun. No, we didn't talk about F2. I'm talking about F1 generation. What is the genotype of F1? This is the first generation that I'm getting. I'm not asking about second generation. First generation. What is the genotype of first generation? Very simple question. I didn't talk about the ratio. I didn't talk about anything. I just asked about genotype. 1:1. I didn't talk about the ratio. Baba, genotype is going to be 1 only. All of them are capital T, small t. This is the genotype. This is going to be the genotype. Yes, yes. Pawan said capital T and small t. Okay, fine. It's okay. It's okay. You know, you are making mistake over here. That's the best thing to do. You make mistakes over here. All the mistakes. You do it over here. Don't do it on the examination day. When you do mistakes over here, I'll correct you. That's the maximum thing that could happen. Okay. There is a dialogue in Hindi. Okay. Kaun char log. Who are those 4 people who are going to see your mistakes? Okay. Who are going to judge you? Do they know you? No. Okay, do they have anything to do with you? No. So keep on doing mistakes, no problem. Okay, so over here, phenotype motivational said, yes, exactly. Priya said it is going to be tall. Why it is tall, Baba? Because this capital T is dominant over the small t allele out there. This is the phenotype. Now Mendel was like, just think like Mendel, okay? Mendel, when he crossed tall plant and dwarf plant, he saw all of them were tall. He was like, Macha, where did the dwarf go? I actually crossed that also. No, where did it go? So he came up with a conclusion that there is a law of dominance. There is something that could be law of dominance, but he was not sure about it, which is why I'm not going to talk about it right now. Let me finish this experiment. He said that, okay, in that case, let me do one thing. I am going to cross between the progenies. This is known as selfing. What do you call it? Selfing, crossing between the progenies. Okay, so over here, again, I have a parent this time. The parents are what? Capital T, small t. Here also it is capital T, small t. Baba, it is homozygous or heterozygous? The parents which are there, they are homozygous or heterozygous? Any idea? Homo or hetero? Heterozygous. Perfect. They are going to be heterozygous. Both of them are going to be heterozygous. Why? Because both the alleles are different over here. Cool, 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 cool. Now let's talk about the gametes. The gametes over here are going to be different. Few gametes are going to be having capital T. Few gametes are going to be having small t. That means few gametes are going to have the chromosome that is carrying capital T. Few gametes are going to have the chromosome carrying small t. Similarly, over here also you get capital T and you get small t. Agreed? Agreed, ma'am. Now we are going to, yes, Priya also heterozygous, perfect. Now we are going to go with F2 generation, the next generation that we are having. Okay, when you cross this, let's see what is going to happen. When you cross this with this, capital T, capital T. When you cross this with this, I get capital T, small t. When you cross this with this, always write the capital letter first, so I'll write capital T, small t. When you cross this with this, I get small t, small t. Now over here, if I see everyone, almost everyone looks different. 2 of them look same, but rest everyone looks different, right? Yes. Which is why I cannot ask you what is the genotype. Rather, I will ask you what is the genotypic ratio. I'll use the term genotypic ratio. So what is going to be the genotypic ratio? The genetic makeup ratio over here. So I think motivational gave me the answer. Priya gave me the answer 1:1, 1:2:1 before, and Pawan gave me the answer right now 1:2:1. How did you get that? For those of you who do not understand, let me tell you. You have been asked about genotypic ratio. Capital T. Do you see anywhere else? No, only one. So I'll write 1 and in bracket I'll write capital T, capital T. Then I see 2 of them which are going to be capital T, small t. Then I see 1 of them which is going to be what? Small t, small t. So what is my genotypic ratio? Genotypic ratio, as Priya said, as Pavan said, it is going to be what? 1:2:1. And whenever you write that, I'll request you from my end, being a teacher who have corrected papers, please put them in brackets. Okay, what do you denote by 1, what do you denote by 2 and everything, please put in bracket. You're going to get brownie marks out there. Okay, yes. Now I'm asking you the next question. What is the phenotypic ratio? Again, I cannot ask you what is a phenotype because they have multiple phenotypes. I'm asking you what is a phenotypic ratio? Come on. Phenotypic, physical appearance ratio. 3:1. Exactly. Priya said 3:1. Pavel said 3 tall and 1 dwarf. Exactly. So basically, 3 of them, if you see, are going to be tall. This is going to be tall. Why? Because both the alleles are dominant. This is going to be tall. Why? Because this is going to be dominant over the recessive one. So basically, I can say 3 of them are tall. 1 papa guy is there, which is going to be dwarf. So I come up with 3 tall and 1 dwarf. 3 tall and 1 dwarf. This is going to be what? The phenotypic ratio. Always, always, always remember this. Okay? This is very important. This can be asked and things like that. Now, Mendel, when he saw this, he was like, now that's what I said, that how can the dwarf feature go? Dwarf feature could have been there, probably would have been suppressed. That's why I could not see that in first generation, but in the next generation I am seeing the dwarf feature. I am seeing the recessive character out there. So he came up with 2 laws. Okay, there are 2 laws that he came up with. One was law of dominance. Can you guess the name of the next law? One was law of dominance. Can you guess the name of the next law? Apart from law of dominance, there is one more law that you have to remember. Law of segregation. Exactly. Pavan says law of segregation. Priya also says law of segregation. And Pavan says law of purity of gametes. Yeah, that is also true. And guys, those of you who are watching this video, Papa, come and comment and try to give the answer. Okay, so this is known as law of segregation and law of dominance, and I'm going to talk more about it. So law of dominance plays a major role over here, claiming that only one particular character is seen in the first generation. The reason being that, or one particular trait is seen in the first generation. The reason is that whenever a pair of dissimilar factors come like this, they are dissimilar factors. Factors are basically genes only. They are dissimilar genes out there. You call them as alleles. Whenever that comes, one is going to be dominant over the other one, which is why you will not see the recessive character, or you will not see the recessive trait in the F1 generation. Okay, that's when he came up with law of dominance, saying that this is the reason why I am coming up with law of dominance, because I did not see any recessive trait in the first generation, because in a pair of dissimilar factors, one basically dominates over the other. Then he came with law of segregation, which he came into conclusion after doing the entire experiment. He saw that I am able to see the dwarf character only because of one reason. That is, whenever gametes are formed, okay, whenever gametes are formed, each of the alleles which are there, each of the alleles, for example, this allele which is over here and this allele which is over here gets an equal opportunity to go into each of the gametes. It is not like capital T allele will only go. It gets an equal opportunity. 50% chance is there that the gamete can contain capital T. 50% chance is there that the gamete can contain small t. Since you have got a chance over here and here, that's when they come together and you get small t allele. So law of segregation says that whenever you are going to form the gamete, okay, there are equal chances for a pair of alleles to separate and move into the gametes. This is law of segregation. It says that whenever you form the gametes, the chromosomes, or basically the pair of factors, are going to segregate or separate and move into each of the gametes. Cool. These are the 2 laws. Please not forget it. If you have a doubt, ask me right now. If you do not have a doubt, give me a thumbs up. If you do not have a doubt, give me a thumbs up. But always remember, after doing the monohybrid cross experiment, he came up with these 2 laws. Only 2 laws. There is one more law that he came up to with the conclusion after doing the dihybrid cross experiment. Is it clear? Do you have a doubt over here? Do you wish to ask me? You can ask. Perfect. Okay, so Priya gave me a thumbs up. So I believe it's super clear for Priya. And Pavan Baba, I hope it's super clear. Krishna Holic, clear? So I have a question over here. I request you guys to solve it. Okay, I have a quick question over here. This is the KCET question itself, and I'll request you to solve it. It goes like this. In one of the hybridization experiments, a homozygous dominant parent and a homozygous recessive parent are crossed for a trait. Okay, they are going to be crossed for a trait. What are the findings that you're going to see in this particular thing? If you're crossing one homozygous parent, okay, dominant parent and homozygous recessive parent, what do you see? Dominant parent trait appears in F2 generation and recessive parent trait appears only in F1 generation. First one option. Second option, dominant parent traits appear in F1 generation and recessive parent trait appears in F2 generation. Okay, next one, okay, someone said C, Pavan said C. Dominant parent trait appears in both F1 and F2 generation and recessive parent trait appears only in F2 generation. Okay, D, dominant parent trait appear in F1 generation and recessive parent trait appears in F1 and F2 generation. What do you think is the answer for this particular question? Pavan says C. Anyone has a different answer for this? I shall move. Anyone has a different answer for this particular question? Okay. Priya also says the answer for this question is going to be C. Yes, Baba, it is going to be C. Why it is C? Here it says dominant parent appears in F2 generation. In F1 you don't see it all, you see not all. So that is why. And recessive parent appears only in F1 generation. Arre Baba, recessive parent, do off. You do not see in F1, you rather see it in F2. So this is going to be a wrong statement. Dominant parent trait appears in F1 generation. Okay, and recessive parent trait appears in F2 generation. Dominant parent you're going to see in F1 and F2 both, but here only F1 is mentioned. That is why I'm going to keep it on hold. The next statement says dominant parent trait appears both in F1 and F2. You saw tall progenies in F1 and F2 both, correct. And recessive parent trait appears only in F2, that is also correct. Dwarf was seen only in F2, right? So this could be the answer. Next thing, dominant parent trait appears in F1 generation and recessive parent trait appears in F1 and F2 generation. Recessive parents you cannot see in both generation, only in F2 you can see it, and dominant F1 and F2 both, right? B can be right if we see grammatically, because then they didn't say F1 only. Okay, grammatically yes, but if at all we have options like this, if we have an option that is talking more in a better manner about it, it is clearly mentioning appears in both F1 and F2, then you should be ideally going ahead with this, right? So if we go like this, probably we might get stuck over here. If you're in a hurry, we might say that this is the answer, but this is why it is said that you have to read all the options. If we have a more accurate option, then basically you can go ahead with it, right? So yes. Ashish, still when can we download verification slip of UG NEET? Dear, they have not given a date, but I'll request you, if the server is fine, make sure you do it quick. Okay, so anyway, the verification period is there until some time now. 8th they might extend it is what I believe. So yes, until 8th or 9th you can possibly, or beyond that, a little beyond that, you can go ahead and download the verification slip. If there is any server issue, they will extend the dates, don't be worried. Okay, so yes, this is not the answer. Okay, this is the answer. Okay, anyways, now there is one more question. This is a homework question. You don't do it right now. This is a homework question. You take a screenshot of this particular thing fast, and once the live ends, put it in the comment section. Not here, put it in the comment section. Okay, yes. Now all about Mendel's law of inheritance. This is what I was talking about. It goes like this. So Mendel proposed that something is stably passed down from parent to offspring through gametes over generation. He called these entities as factors. What do you call this right now? You call this as gene. Something is being passed. What is this something? That is basically the gene itself. Genes are unit of inheritance. Gene carry the information required to express specific traits. Genes which are there, they're nothing but the segment of DNA, and they code for protein, and those proteins are responsible for each of our character. Okay, alleles are different forms of the same gene, different type of a gene that we have, they are known as alleles. Okay, he proposed that in a pair of dissimilar factors, one dominates the other and hence is called the dominant factor, and this one is known as the recessive factor. Done? Yes. He also said during meiosis, allele pairs segregate and only one is passed into each gamete. So when meiosis happens, as I said, this chromosome and this chromosome is going to be segregated and only one is going to pass down into each of the gamete, because in meiosis, if at all you remember, half of the chromosomes are going to go. Right? Right. Cool. Now over here, this is the law of dominance. Law of dominance says, please write it because this can be a second PUC question. This has been a second PUC question. Characters are controlled by discrete unit. What do you call them as? Factors. Factors, they occur in pairs. You know that in a dissimilar pair of factor, one member dominates over the other one. Yes, yes. The law of dominance is used to explain the expression of only one parental character. So if at all you have been asked, which is that law that explains that only one parental character is being expressed in monohybrid cross in the first generation? That is law of dominance. Please make sure you write it and you explain about all of it. Okay, then there is law of segregation, which goes like this. I'm just reading it out because I explained it already. Alleles do not blend. Both of them appear unchanged in F2. Alleles which are there, they are not going to blend with each other. Capital T, small t, they are blending and giving a middle character. No, that is not happening. They are going to go exactly like that. The chromosomes are going to exactly go like that, and basically depending upon the combination that is there, we are going to see a character. During gamete formation, this is important. During gamete formation, allele pairs separate, so each gamete gets only one allele. This is law of segregation. Homozygous parents produce identical gametes, means same gametes. Homozygous, they are going to produce same gametes only. Okay, whereas heterozygous parents which are there, they are going to produce two different type of gametes. This is also explained by law of segregation. Okay, yes. Okay, now we go ahead with the next thing which is there, but before I go ahead with that, I have to answer, guys, just excuse me for a minute. Sakiba, I told 5:30 I'll answer her. I'm late already. Just let me answer her question if I know the answer. Ma'am, suppose a student has got 3K rank but the college cutoff is 2K. Okay, he has not allotted this college in the first round. Does it mean that there are no seats are available in that college? If he is allotted the college in the second round or third round, why he has not allotted a seat in the first round? Does there a rule that these percentage of seats are allotted in the... Okay, Sakiba, is because, listen, in the first round you probably might have not been allotted with the college because the people who come under that particular rank range will be getting that, and mostly that rank range is the top colleges, they just fill it up. I'm talking about the top colleges. Okay, if at all those seats are being filled up, it's because the people who are writing JEE examination, things like that, they are awaiting for their results. Now they're not very sure about the results out there. They could have been not sure about the JOSA results out there, which is why they held this particular seat. But now, since round 1 has been passed since long time, now you're going to round 2, there are chances a lot of them could have cancelled their seat. They might have cancelled their seat, which is why there will be seat availability in the next round, that is round 2. You get it? And for the lower college, for colleges that are not that great, children who come under the rank range and they feel that it's I might not get any college, they will pick it up in round 1 itself. In that case, there are lesser chances for you to get the same college in the next round. But sometimes one more thing happens, since they have got a college, what do you say, not that great college in the first round, they might also skip it and go with management seats, and you might get that particular seat also back in the seat pool. It's because of that. It's not because of percentage of things like that. Whatever number of seats are there, whatever children are there, that is going to be filled up in each round. Okay, Sakib, I hope I was able to answer your doubt. Yes, now I'm going back to my class. Okay, and if any more doubt is there, you can comment it down. I'll gladly reply to it. When will the seat matrix will be released before the second round? It might take time. Okay, because the NEET counseling and everything is going on right now, and NEET children are also participating, which is why they are super busy, I think, with all of these things. They might release it very, very soon, or it might take time. I think by mid of August. Okay. Redmi, you have to do verification, document verification process now, so it's over for, you're talking about UG NEET, then it's different. The UG NEET verification is still open, you can check it out. But for KCET, it's been closed already. You can call KE and check that up once. Okay, now my dear children, coming back to this particular thing. Okay, so is there any easy way to cross this? So there is one person who is saying, yes bro, there's an easy way. And by the way, my name is Reginald C. Punnett. I am a British geneticist, and I know that's too much information about me. Well, listen to me. Reginald C. Punnett is a very important person, a very important character, and you have to remember that he's from British. Okay, so he's from British. He's having a British origin. So I am a British geneticist. They can ask you German geneticist or whatever things are there. Always remember, he is a British personality, and his name is Reginald C. Punnett. He came up with Punnett square, and Punnett square is actually very, very easy in comparison to the crossing thing. In Punnett square, what do you do? Suppose you want to cross this and this. What do you do is that you make the square, you put two alleles over here, small t, you put two alleles over here, and then you cross. This and this cross, you get this, this and this, you get this, this and this, you get this, and finally this and this, you get this. So this becomes more easy when you have multiple other characters also being spoken about at the same time. Anyway, please remember Reginald C. Punnett. Okay, now came Papu. Okay, this is Papu, by the way. You're going to see Papu throughout this particular year. Papu is my friend. So Papu says, ma'am, I have a doubt over here. How did Mendel get to know if the plant is tall like this or like this? So Mendel saw a plant, it was tall. How can Mendel say whether it is like this, homozygous tall or heterozygous tall? Do you have an answer for that? Do you have an answer for Papu's question? If yes, you can go ahead and try to answer. If yes, you can go ahead and try to answer. Test cross, ma'am. Pavan says test cross. Okay. What about others? Test cross is something that you want to do for finding out whether it is going to be homozygous tall or heterozygous tall. Okay. Papu is like, okay, Pavan, let me check. I'm going to believe only if Jasmine Ma'am says that. So listen to me carefully. Papu, what Pavan said is actually true. Test cross is the actual answer. Okay, what do you mean by test cross? That is even more important. Test cross definition is very, very important, and you can cross it and show it as well. Typical test cross is where a plant showing dominant phenotype is crossed with a recessive parent instead of self-crossing. Whenever you have a dominant phenotype means physical nature. What is the dominant phenotype in tall and dwarf? It is going to be tall. It is going to be tall, right? It is definitely going to be tall. Now, a plant that is showing a dominant phenotype, to understand whether it is, whether it is going to be tall like this or like this. Anyway, because it is showing what? The dominant phenotype. And what is the dominant phenotype over here? Dominant phenotype is tall. And to just be sure about whether it's this or this, I am going to cross it with a recessive parent. Always understand, I will be crossing it with a recessive parent. And what's the recessive parent over here, Baba? It is going to be small t. Sorry, just a second. I did a mistake. Yeah. What is recessive parent over here? It is going to be small t. This type of cross is known as test cross, and you are going to understand its importance right now while I talk about it. So suppose over here, I do not know whether the plant is tall like this or like this, which is why I am going to do what Pavan is telling me. Test cross, okay? So I am going to take my plant. Suppose the plant is like this, and I'm going to take the recessive parent plant like this. Okay, let me see what I get. When I cross them, I see all of them are going to be like this. Capital T, small t. Capital T, small t means all of them are dominant over the small t allele. All of them are going to be tall. All of them are going to be tall here in this particular case. Okay, now here if at all we look, you have capital T and small t, and now you're going to cross it with the recessive parent. Now you're going to cross it with a recessive parent. So you have small t over here, and you have capital T small t over here. This is where you're going to see the difference. Check it out very, very carefully. When I cross this with this, I get capital T small t. When I cross this with this, again I get capital T small t. When I cross this with this, I get small t small t. When I cross this with this, I get small t small t. You look at this and tell me, are all of them tall? Are all of them tall over here? What do you think? Yes or no? No, exactly. Both two of them are tall and two of of them which are there, they are going to be, sorry, 2 of them are going to be tall and 2 of them are going to be dwarf. So ratio is going to be 1:1. So whenever I cross the plant with the recessive parent and I get something like this, I understand that the tall parent which is there, it is tall plant which is there, it is actually like this. Whenever I see all of them are going to be tall in the progeny, I get to understand the tall is actually tall like this. Am I not able to understand whether it is homozygous or heterozygous? I will be able to understand. And why do you think this is important for me? This is important for me because suppose I am a farmer and I want all the plants to be tall. In that particular case, I will make sure that I take a homozygous tall over here. I want a homozygous tall plant over here. Okay, that will be more easy for me. Otherwise, I will be seeing dwarf plants later on. So that's when it becomes more important. This is just for giving you an idea about things. But remember, this is what you mean by test cross. Test cross is when I have an organism that shows a dominant phenotype. I want to find the genotype of it, whether it's homozygous or heterozygous, which is why I am going to cross it with a recessive parent. It has to be recessive. If it is dominant over here, the dominant trait is going to be appearing because dominant are always dominant, right? So you need a recessive parent over here to be crossed. Got it? Yes, Pawan, 50% tall and 50% dwarf. Did you get this particular concept of test cross? If yes, can you quickly give me a thumbs up? If everyone has understood it clearly, just quickly give me a thumbs up. Yes? No? I'm waiting for your thumbs up, by the way. Yes, perfect. Cool. Now, there is one more term that I want to discuss over here, and this is very, very important. By the way, this is the example that is given in NCERT. I'll come back to that. Back cross. What do you mean by back cross? Okay, so back cross and test cross is almost same. Back cross means you're going back and fusing with either of the parents. It can be recessive, it can be dominant. Okay, back cross means you are crossing an F1 hybrid, the one thing that we have got in F1 with basically either of the parent. Can be recessive parent or it can be dominant parent. I do not mind. Okay, specifically when you're crossing with recessive parent, you now call this term as test cross. So back cross is a broader term where you're crossing with F1 hybrid with either of the parents. Test cross means when you're specifically crossing with recessive parent, you have a term for it, and that is known as test cross. That's the only chutku thing which is there. Just remember it, it could be helpful for you. Now the example that is given in your NCERT, it looks like this for the test cross. Okay, so there is a violet flower. We do not know whether the violet flower is like this, homozygous violet, or it is heterozygous violet. By the way, let me tell you, violet, V starts with V, right? So it be denoted by V, capital V, capital B. But this is a trait which is denoted by W. Do not be worried. Okay, it's not the case for all the traits. Anyways, so capital W is there, small w, capital W, small w is there. What do? I do the test cross. For that, I have to cross it with what? A homozygous recessive parent. A recessive parent. So I'm going to cross it with this. When I cross this with this, I see all of them are capital W, small w. That means it is going to be dominant over this. My plants, all of the flowers are going to be violet. Okay, if they are violet, I understand it is like this. If all of them are violet. On the other hand, when I cross this one with this, what I see, 2 of them are violet and 2 of them are white. 1:1 ratio. Got it? So just have an understanding of it. I had already explained it, which is why I'm not going to go ahead. Now, until this particular point, is there any doubt which is there? Any single bit of doubt that any one of you is having? If no, I can go ahead with the next thing which is there. There are some questions that I want you to solve, and not just questions. I want to go ahead with exceptions of Mendel's law. Yes, ma'am, clear, perfect, thanks Macha. Okay, so here is a question that we all can solve. This is 2024 question from this chapter, 2 years back. Test cross in pea plant is what? A cross between F2 tall plant and recessive parent, a cross between F2 dwarf plant and recessive parent, a cross between F2 tall plant with dominant parent, a cross between 2 F1 plants. What do you think is the answer for this particular question? A, ma'am. Pawan says A. Okay, what about others? Priya? Sakiba is also saying A. Oh ho! Sakiba is a senior right over there. Sakiba also says A. So A is the answer. Shall I cross A? Yes, A is going to be the answer. Test cross in pea plant is what? A cross between F2 tall plant and a recessive parent. Cross between dwarf plant and recessive parent, that you do not have to do. Okay, cross between F2 tall plant with a dominant parent, you cannot do that. A cross between two F1 plants, that is also not needed. So this is going to be the answer. Okay, they can mention F2, they can mention F1 over here. Always understand, if they are mentioning dominant character over here and they are mentioning that you are crossing with the recessive parent, that is going to be correct. Okay, motivational. Priya also gave me the correct answer. Vasudev is also there. Vasudev also gave me the correct answer. Perfect, perfect, perfect. Lots of love. Okay, to all my machas and machis out there. Now going ahead with exceptions of Mendel law. This was the first question that I had. Anyways, exceptions of Mendel law. As I told you, one very important thing, that is, what was Mendel expecting, okay, when he crossed red color flowers and white color flowers, and I told that red is dominant one. We all expect one thing, that it is going to be, the progenies are all going to be red. But in this particular case, it was different. Dhum tanana is happening over here. Okay, so there is something which is known as incomplete dominance that we have not seen until this particular point. And I'm going to show you first, and then we are going to see what is exactly happening over here. So what do I do is, I cross a red plant, okay, with what? White ones. I know R stands for red, and small r which is there, it stands for white. Which is why I believe that since R is capital letter, I believe that it's a dominant trait, right? Right. Now when we make the gamete of it, by the way, these are the parents. Okay, now when I form the gametes, capital R and capital R is going to be there. Here I get small r and small r, right? These are going to be the gametes. Okay. In a gamete, only one chromosome passes from here, here. Similarly here, here. Both of them cannot go to one particular gamete out there. Now when I cross them, let's see what is going to happen. This is going to be the first generation. When I cross this with this, I get capital R, small r. When I cross this with this, I get capital R, small r. When I cross this with this, capital R, small r. When I cross this with this, I get capital R, small r. Now I have a question to all of you. First question, listen to my question carefully first. Papa gave me the answer already, pink color. Yes, exactly. Okay, so my first question was, what is the genotype? But anyway, since you have told me the phenotype, what is the physical appearance that this plant is going to have? Ma'am, it is going to be red. Some people might be thinking in their head, they are not saying it aloud. It could be red because R is dominant over the smaller. But ma'am, you said there's an exception, so probably something else? Yes. And as Priya said over here, the phenotype is going to be pink in color. Mendel was like, ooh la la, what is happening? Actually, it is not Mendel. Someone else did the experiment and he was claiming that Mendel was wrong. Mendel bhai sahab told na that one is dominant, another is recessive, so it should be having red in color. But why it is pink? Mendel is wrong. Mendel did something very, very wrong. He was claiming that. Okay, he was claiming that. So then this person, for a surety, he thought that, why not? Let me cross it once more. Let me do the same selfing that Mendel did and let me see what is going to happen, just in case if I find something else before blaming him, right? So about the genotype first before I go ahead with that. Genotype is going to be capital R, small r. Okay, now I'm going to do the selfing. Listen to me very carefully. These are the parents. I have capital R, small r, and I have capital R, small r. What are the gametes over here? Gametes are going to be capital R and small r. Here also capital R and small r. Now I'm going to fuse them. Okay, I am going to fuse them. Let me fuse them for F2 generation. When this and this comes together, I get capital R, capital R. When this and this comes together, I get capital R, small r. When this and this comes together, I get capital R, small r. And when this and this comes together, I get small r, small r. Ma'am, capital R, capital R, capital R, small r, this is same like how we had for tall and the dwarf ones, right? Okay, now I'm going to ask you one thing. What is going to be the phenotypic ratio? I'm asking you one thing. What is going to be the phenotypic ratio? Motivational says capital R, small r is a genotype. Yes, yes, motivational says capital R genotype. That is correct. I'm asking another question over here. This is F2 generation and I'm asking you a question. What is the phenotypic ratio over here? At this point, what is the phenotypic ratio? Pavan says 1:2:1. It should be 3:1, no? It should be 3:1. How can it be 1:2:1? How can this be 1:2:1? I don't get it. Saqib also says 1:2:1. Motivation also says 1:2:1. Since you're saying I have to agree this, listen to me very carefully. Until 3:1 ratio, unlike 3:1 ratio that we saw in Mendel's experiment, here you get to see 1:2:1 ratio. How do you get that? Listen, this is going to be red in color since both of them are capital R, capital R. These two things are there, capital R, small r is there. Okay. Capital R, small r is there. You saw that capital R, small r was pink, which is why here also it will be pink. Small r, when it comes, it is going to be white in color. No issues with that. Which is why I'll say 1 is going to be red, 2 of them are going to be pink, and 1 is there that is going to be what? White in color. You are going to see 1:2:1 ratio in this particular case, that is phenotypic ratio. Okay, ma'am, got it. Okay, so that means Mendel was wrong over here. Agreed? Agreed. Okay, let's find one more thing, genotypic ratio. Let's find the genotypic ratio. Guys, come on, tell me, what is the genotypic ratio over here? Genetic makeup. 1 capital R, 2 capital R, small r, and 1 small r, small r. Sakiba says same. Okay, easy, shortcut, easy. Same. Okay, Priya also has the same answer, is it? Yes, Macha, it is going to be the same only. Right, it's going to be the same. One of them, genotypic means genetic makeup. Only one looks like capital R, so I'll write 1 capital R. Okay, 2 of them basically are capital R, small r. And one of them which is there is going to be smaller. So genotypic and phenotypic ratio for the first time is going to be the same, Macha. For the first time is going to be same, and Mendel over here is going to be wrong. Mendel over here is going to be wrong. Right. So people started claiming that Mendel's work was not correct. For 7 years, that guy worked for no reason. This was a backlash that he got. I told you in the initial part of chapter, whenever we do things, we will get backlash. Okay, that is something that is there in human tendency. We are going to get backlash, but one day, if you had put in hard work, it is going to be recognized. Okay, so yes, you are seeing the backlashes that Mendel was facing at this particular point of time, but you will see in this chapter a point where he was proven to be actually correct. So my dear children, without waiting until that particular time, I'll tell you one thing. So there are exceptions. There are cases where Mendel's law were not being followed. But it's not always like that. There are cases where Mendel's law were being followed. So he was not wrong over there. He was correct only, but he didn't talk about the exceptions. So yes, you see an exception right over here. This exception is known as incomplete dominance. What do you mean by incomplete dominance? It means that this particular dominant allele is not completely dominant over the recessive allele which is there. It is not able to be completely dominant over what? The recessive allele which is there, which is why you will call this as what? Incomplete dominance. And where do you see it? You see it in Snapdragon or Antirrhinum majus. Where do you see it? You see it in Snapdragon. Snapdragon which is there is very, very important, and Antirrhinum majus is also important, the same plant only. You also call this as 8 o'clock. Sorry, what do you call this as? 4 o'clock plant. Guys, just give me a second. I'm getting a call. Just a second, okay? Important call, I feel. Yeah, bro, tell me. Huh? It's his number only, da. I'm in live right now. Yeah. Okay, so sorry guys. Yeah, but he is dead, and before he was proven right now, ma'am. I hope so. Yes, I did not check when he died, but I hope so that when he was alive was proven right. It took 37 years for him to be proven right. Don't worry. I mean, that was there. 37 years is a pretty long time. I don't think it was during the time he was alive, but yes. But at least we know about them here at this particular point. I hope no one has to wait that long for getting proven right. So this is a story that I am building. Listen to me carefully. It's not like he was getting too much backlash then and people were going against him and things like that. This is just to make things interesting. Obviously, there were times where he was proven wrong, and there was no media like this where someone would come in the television and say that this person did wrong. There was nothing like that. There were definitely other modes of communications as well, but yes, it was not as bad as we have in today's world. Okay, so don't be worried. I know it's so sweet to see your little hearts being sad about it, but don't be worried about him. I think he was fine with it. Yes. Okay, so this is incomplete dominance, and I hope you remember the name of snapdragon and antirrhinum. The question, whenever MCQ is asked, it has been asked on this particular name, and you are like, what is antirrhinum? I do not know it. So antirrhinum majus is the same thing, that is snapdragon, and over here you see the plant. Okay, this is how it looks. This is how it looks. Okay, ma'am, also in 4 o'clock plant. Yes, the same thing. 4 o'clock plant, the same name. Snapdragon, 4 o'clock plant, antirrhinum is all the same thing itself. Cool? Yes. Is this portion understood? Incomplete dominance is clear to all of you? This is the first exception that we have. By the way, to read it, incomplete dominance is a type of inheritance where neither allele is completely dominant. Neither of them is completely dominant over the other. As a result, the offspring shows a blending of traits. Offspring is going to see a blending of traits, and you have to make sure you make everything like this. Don't draw the diagrams. Make the genetic makeup and stuff like this, such that you get full and full. Okay? Done? Deal? Yes, crystal clear. Thank you so much. Now we are going to go ahead with the other exception that was there, and that was multiple allelism. Okay, now this is again a little difficult, a little tricky to understand, but you will understand it. Okay, not too difficult. So until now, Mendel said that usually over here you have two alleles for height, right? One is capital T, one is small t. He never mentioned about multiple alleles for one particular character. This was a character height, and for that he said there were two alleles. Whatever he studied, he said there were two alleles for that. He never mentioned, or he went beyond that particular time. Oh, this is the last topic for today then, ma'am. Okay, so I was actually planning to take the class until 6:30 today, because I really want to cover the major portions of it. Whatever we can cover until 6:30, it will be done, and after that I'll stop it. Okay, just 25 more minutes. Perfect. Okay, so yeah, because I think this should be going in continuation. Anyways, so people came up and said that it's not always like that. One character can have multiple alleles. What are you saying? Only two alleles cannot be possible? There are characters with multiple alleles. Example, blood group. In blood group, Baba, you have three different alleles: IA, IB, IO. What do you mean by that? Basically, I mean that here when I say there's a gene for my blood group, this gene can be IA. This gene can be IO. In another case, basically, it can be here, it can be IA. Here it can be IB also. There are multiple alleles. A is there, IO is there, IB is there. There are multiple alleles for this. So when there are multiple alleles governing a single character, you call this as what? Multiple allelism. You call this as multiple allelism. Are there an example of multipleism? This is the example, that is blood groups. Now I'm going to talk more about it. Listen to me, okay? Because here, talking about blood group, they show two exceptions. One is multiple alleles and one is co-dominance. Until now, we saw that one is dominant, other is recessive. Have you ever seen but both of them are equally dominant? We saw two conditions. One is dominant, other is recessive. We saw the next condition where both of them are not dominant at all. That's why incomplete dominance is seen. Now in blood group, we are going to see one more exception, that is co-dominance. Two alleles are going to be equally dominant. All of this is going to be seen over here itself. Okay, so here, remember, the plasma membrane of the red blood cell has sugar polymers that protrude from its surface, and the kind of sugar is controlled by the gene. So basically, if this is the plasma membrane of a red blood cell, there are sugar polymers that protrude out from it, and what type of sugar polymers are going to be governed by a gene? And this gene has what? Three alleles: IA, IB, and IO. Sometimes this IO is also mentioned as small I. Okay, it is also mentioned as the small I as well. But there are three alleles. Okay, they are coding what? They are coding the protein that is going to be protruding out from the plasma membrane. The gene I has three alleles. I spoke about it. Yes. And now we are going to see different type of blood group thing. And this is very interesting. I love it. Ma'am, मेरा verification sheet में 12th के marks missing है, क्या करूँ? Dear, while you registered, did you fill in the exact roll number and everything that you had, such that the KA could be fetching in details? If not, I'll request you, Ayush Patel, please call KA once and talk about it. It's very important. You're talking about verification slip. in the verification slip, right? All the other details are there. If all other details are there, then do not be worried, but still call the KA once, because downloading the verification slip is the most important part. If you have done it, then it's all cool. Okay, Ayush, call them once. Cool? Yes. Fine. Now, I'm going to go ahead and talk about certain things. Ready to study? So, here, people can have few blood groups. One is A blood group, one is B blood group, one is O blood group, and there is AB blood group. When does someone get A blood group? When basically one allele is A and other allele is also A, that's when you can get A blood group. Or there can be a condition where one allele is A and other allele is O, that's also when you can get A blood group. How, ma'am? Because A is dominant over O. A is dominant over the allele O. Okay, so there are two conditions where a person can get A blood group. Now the second one is, a person can get B blood group when both the alleles are B, or when one allele is B and other allele is O. How, ma'am? Basically, B allele is also dominant. Ayyo, B allele which is there, it is also dominant over O. With this, I got an understanding that O is a recessive allele in both the cases, and O is going to be expressed only when both the alleles, okay, only when both the alleles are going to be O. Just a second, there's a glitch. Both the alleles have to be O, then only it is going to be expressed, because when B is there, it is dominant, and when A is there, that is also when it is dominant. So you just have one option, and that is this one. Right? Now you tell me, when does a person get AB blood group? I have a question to you, and that is, when does a person get AB blood group? That is very, very rare. Any idea? IA and IN. When does a person get AB blood group? IA and IB, exactly. A person gets AB blood group when both of these alleles are going to be same. When both of these alleles are going to be A and B, that's when it is going to be getting AB blood group. Why? How can it be happening? Here A was dominant over O, here B was dominant over O. Then how can you get a person having AB blood group when it is having something like this? Because, Baba, it shows something that is known as codominance. It is showing something that is known as codominance. Understood? And this is also an exception. Mendel never spoke about codominance. He never spoke that there will be both the alleles who are going to express themselves. That is why it is also an exception. Incomplete dominance is an exception. In blood group, you have multiple allelism. That is an exemption. Codominance is also going to be an exception. Now, you can get questions like this, and you have to be very, very careful over here. What do I mean by you can get questions like this is basically, listen. Okay, in this particular scenario, okay, when I'm talking about codominance, suppose they can ask you, if a person has A blood group, okay, and they are crossed with a person has A blood group, another person has AB blood group, what will be the blood group or the genotypes of the progeny? What will be your answer in that particular case? If I get a question ever like this, that a person has A blood group, another person has AB blood group, what are going to be the genotype of the progeny? What will you say is the answer for this particular question? A and AB. I'm asking about genotype. I'm asking about genotype, not phenotype. How many genotypes are possible in the progeny? Think. I'm writing it here. Okay. So there is going to be A and there is going to be AB. This is the blood group. Now, how many genotypes are possible? So, motivational says, okay, IA, IV, okay. Sakiba says 4 genotypes are possible. Let me check. Okay, let me check. Over here, if I say A, the scenario can be like this. IA, right? AB, it's very simple. It is going to be IA, IB. When I cross them, what do I get? I get IA. This is the first genotype, first type of genotype. When I cross this and this, I get IA, IB. This is second type of genotype. When I cross this and this, I get IA again is the same thing, so I will not count it again. When I cross this and this, I get IA. From this I see two different genotypes are possible. One is this IA, and the other one is this IA. Two genotypes are possible. But will I consider only this scenario? No, I have to consider the other scenario where a person can have IAIO. This is also a blood group, right? And I have to talk about maximum genotypes. So this is also a blood group, and IAIB remains the same for AB blood group. For AB it remains the same. Now I'm going to cross it and check. Okay, when this and this crosses, I get IAIA. First one. When this crosses, I get IAIB. Second one. When I cross this and this, I get IA and IO. Third one, different genotype. When I cross this and this, I get IB, IO. Fourth genotype. How many genotypes am I getting? I am getting 4 genotypes, and Sakiba Baba, you were correct. This is the maximum number of genotypes I'm getting. I will not consider this because I'm getting minimum number of genotypes. If you get ever a question like this with A and B and you have to tell the number of genotypes, you have to talk about the maximum one. Okay, so the answer is going to be 4. And if you are asked what are the phenotypes, the 3 phenotypes, sorry, how many phenotypes are possible? What are the number of phenotypes which are there? Come on, tell me that also. How many number of phenotypes are also possible? Let's talk about that over here. Number of phenotypes, some number of genotypes you saw. Let's talk about the number of phenotypes as well. 2 phenotypes. Pakka? Sakiba says 3 phenotype. Check the phenotype once more. Okay, let me check. Okay, let me check. This is A blood group. Phenotype, physical appearance. This is also A blood group, so I'll just count it as 1. Okay, I'll count it as 1. This is AB blood group, so this is second one, and this is going to be B blood group because B is dominant over. So 1, 2, 3, no, A, B and AB. So there will be 3 phenotypes. Oh yes. So these type of questions could also be coming in there, which is why we have to be really careful. Got it? Perfect. Clear? Akash said 3. Akash, you were correct, macha. Okay, now I'm going to go ahead over here and we're going to solve some questions. I think I have some questions for you. Let's go. A man with blood group A marries a woman having blood group B. The maximum possible blood groups among their progeny are what? Among their progeny are going to be what? Come on, try. A, ma'am. A is the answer. Are you sure? Guys, everyone has to answer. Pavan only answer this question. Chalo. Everyone, try. A man having A, man having A blood group marries a woman having B blood group. Now, the maximum possible genotypes you have to find out. Only one answer I've got, and that is A, which is why probably I have to go ahead and try to find the answer if you are not trying from your end. Only one person said the answer. That is why I'm waiting. Motivational, come on. Chalo. Okay, motivational says D is the answer. So then I'm guessing. Okay, then I'm going to go ahead. Since I have to talk about maximum possible genotypes, I will not consider this. I am not going to consider this because this is not going to help me. Okay, this one does not help me. Whatever I cross, I'll get AB only. So I'll consider this one. So I'm going to consider this one. Okay, IAIO and IBIO, and I'll cross them. When I cross this with this, I get IA, IB. What is the blood group? AB blood group, because both of them are codominant. When I cross this with this, what do I get? IA, IO. What is the blood group? A is dominant over O, ma'am, which is why A blood group. Okay, cool. When I cross this with this, I get IB, IO. What is the blood group? B is dominant over O, ma'am. I'll get B blood group. Okay, I've got 3 blood groups already. Okay, finally, when I cross, where is that? When I cross this with this, IO. What do I get? Baba, there is no one to dominate over O. Since it is recessive character over here, it is going to be O blood group. How many blood groups do I see? A, B, and O. And where do I see that? In A option. Socrates there saying option A is the answer. Exactly. So everyone who said option A, answer is correct. And if you have went with D, I hope now it's super clear for you. Okay? Akash also says it is A. Perfect. Cool. And I'm proud of every one of you, regardless of what answer you give me, because you guys tried. That is very, very, very important for me. Cool. Let's go ahead then. The next question that is there. Yes, ma'am, clear. Little confidence is coming. Come on, try this one. Okay, a genotype of a husband and a wife are IA, IB and IA, IO. Okay, among the blood types of their children, how many different genotypes and phenotypes are possible? Come on. I am saying that husband and a wife. Husband is basically IA, IB, and the other person that we have, the wife who is there, is IA, IO. You have been asked how many genotypes and phenotypes are possible. Everyone. Right. Try to find it out. Cross it. Akash says the answer for this question is going to be C. Okay, let me see if Macha Akash is correct or not. I'm waiting for others also to answer. Pavan says D is the answer. Oh, C and D. I've got C and D option. Anyone with A and B? Are we all solving? Socrates says it is B. Now ABCD option have been given to Jasmine Ma'am. Jasmine Ma'am has to choose from us. Multiple choices for me. Okay. Let me go ahead then. Since motivational, I'm waiting for your answer also, Baba. Equal treatment. So I'm gonna try, okay? 3, 1, 2, 3, let's go. So over here, when I cross this and this, I get IA. Blood group is what? I'll write it over here, A blood group. When I cross this with this, I get IA, IO. What is the blood group? A is dominant, ma'am. Again, A blood group. Okay. When I cross this with this, I get IA and IB. What is the blood group? A and B both are equally dominant, ma'am. So it will be AB blood group. Okay. Then finally, we have IB and IO. B is dominant over O, which is why B blood group. Now, we have to check how many genotypes. IA, do you see elsewhere? No. So this is one genotype that we are having. Okay, one genotype IA. Second genotype A and O. A and O, do I see it anywhere else? No. This is second genotype that I am having, A and O. Third genotype A and B, do I see it elsewhere? No. The third one is going to be A and B. The fourth one, B and O, do I see it elsewhere? No. The fourth one is there. So basically I have 4 genotypes. How many genotypes? 4 genotypes. What is the physical nature? What is the physical nature going to be like? Let's check it out. Both of them are A. I will not count it as 1 and 2. I'll say it is 1. One phenotype A, second phenotype AB, and this is going to be the third phenotype. So 3 phenotypes and 4 genotypes. 3 phenotypes, you have 3 phenotypes, and 4 genotypes. B is going to be the answer for this particular question. Dun, dun, dun. Why not other options? Because here 4 phenotypes is given. You do not get 4 phenotypes. This is the same thing only, no? So you do not count it 4. Okay, you counted 3. Genotypes, what do you see? They are different. Genetic makeup is going to be different in all of them, which is why we say 4. Which is why the answer for this question is going to be what? B. Got it? Little confusing topic, I know, but you're going to get it while solving questions. Okay, it is going to be okay while you solve the questions. So don't be worried. So, clear? If clear, give me a thumbs up. Don't be disheartened, please. Remember, Mendel was a person who didn't give up, right? Motivational says, okay, perfect. So don't give up. Try to solve as many questions as you can. Chalo, I'm going to give you one more question. This is 2021. By the way, all of them are KCET questions, okay? All of them are KCET questions. Yes. What is a possible blood group of children whose parents are heterozygous for A and B blood group? Come on, let's go ahead. Last one, and this one, don't take your time. Please take your time over here. Don't give me the answer firstly. Try to write it, try to check, and then give me the answer for this, okay? What is the possible blood group of children whose parents are heterozygous for A and B blood group? Heterozygous, not homozygous, they're heterozygous for the A and B blood group. Come on. And please take your time. Motivational says B is the answer. So motivational gave me the answer after. Okay, B, ma'am, same as 2020 PYQ. Okay, B only. Socket also says it's B, and motivational also says it is B, which is why probably I might have to log B. Do I have to? Pavan. Let's wait for Pavan also. Okay, since Pavan was there since this long. Okay. When I say heterozygous for A and B blood group, when I say heterozygous, it is going to be IAIO. If I said homozygous, it would have been this. But this is not the condition. That has not been asked over here. You have been asked about heterozygous condition. So this is the one. The other one for B, then it will be IBIO. Now when you cross it, what are the answers going to be like? Let's check it out. This and this, you get IAIB. That means AB blood group. Phenotype is AB. When this and this is crossed, same thing. IAIO, it is going to be A blood group. When this and this, it is going to be IBIO, that means B blood group. And finally, this and this, IOIO, it is going to be O blood group. Phenotypes, basically, what are the possible blood groups? That is basically A, B, AB, and O. The answer for this question is going to be B. Got it? Clear? If this is super clear, give me a thumbs up. If it's super clear to all of you, give me a thumbs up. And if you have any doubt that you want to ask me, please do ask me. Because if you do not ask me, I do not know whether you have doubt or not, right? So that's why I'm telling you. Okay, perfect. So I've got my thumbs up, and I think I'll end up my live over here. Do you have anything else to say? By the way, I want to tell you something. Don't go, it's an important thing. Okay, so basically under this particular video, you will be able to see something like this. Join the free ACE KCET series. This is something that is known as all-in-one page. Okay, basically each and every classes that we take is going to be reflected over here, and that too subject wise. So if I do a one shot, okay, you do not have to see the recorded session. You will be able to see that over here. It's linked to YouTube, and you don't have to pay anything. If you go with buy now, basically it will be like asking you your phone number and things like that. That's all. There won't be anything else that is going to be asked, and you are going to see subject wise segregation, that each subject, okay, each subject, biology, physics, maths, everything is going to be like that, and under that everything will be there. Assignments will also be soon given to you guys, so that you can go ahead and practice it. We are working on it, but it is definitely going to be helpful for you. The site is ready, all the videos are there. You can check it out if at all you have missed any session out there. Okay, so do check it out. That is known as all-in-one page. And one more very important thing, I request every one of you to join the WhatsApp channel. Now in the WhatsApp channel, there are almost 31,000 people out there. Those are your seniors as well. Those people are going to soon exit the WhatsApp channel. Why? Because their counseling process and everything is going to be over. So yes, now it's time for you guys to come in that, and over there you're going to get all the updates regarding what all sessions are going. Whenever live comes, we put messages out there that we are coming live for the session, so you're going to get updated about it. That is also be there in the same particular thing. So if you move down, no, this is KCET WhatsApp channel. You can just click on this and join the WhatsApp channel. Okay, And I mostly message up there in the WhatsApp channel, even though it's not fully handled by me, but I try my maximum to get in touch with you guys up there as well. So make sure you be there. Okay? Ma'am, your fluency is very good. Thank you so much, Amp. Okay, thank you so much for the wonderful session, Pawan. Thank you so much. And ma'am, I actually got 44K in need, and I'm really thankful for all the guidance. Thank you so much. That's so sweet of you. I really wish to take a screenshot of all this thing. Ma'am, I'm unable to register for Karnataka counseling. Can you please help me up? Dear, you have to go ahead. What's the reason? Why are you unable to register for it? Do you not meet the eligibility criteria, things like that? Are you not getting the, what do you say, the link out there, or is there any other issue? Pavan is asking, you ate, ma'am. That's so sweet of you asking to me about that. I had my lunch. I hope you also had your lunch. For dinner, I have to go home and probably cook something. Yes. I think there is a site issue. Okay, in that case, just wait for some time, and as I said, please keep on trying. That is very, very important for you to try. If you do not try and then later ask you, it will be a problem. Also, simultaneously, I'll ask you to just call them at once and ask. I know it's very difficult for you to get through that particular call, but please go ahead. Okay, ma'am, I got 25,000 in need. Which college could I get? Peaceful, I do not remember the college floor, dear. I really have to check that. I'm so sorry for disappointing you. Okay, if it's possible, I'll definitely try to make a college-wise, rank-wise cutoff out there. Okay, not promising you, but I'll try. Okay. So I think that's all, children. Since there are no other issues, by the way, everyone has their FA examinations been done, right? The FA examination has been done. Motivational, I know for you it's later on, but for others, I hope FA1 examination is already done, completed. Is it possible to communicate with you so you can explain the issue in detail? Yes, there's only one way to communicate, and that would be YouTube, according to the contracts that we are having with the company. So, yes. No, dear, today I can rest peacefully. Okay, then thank you so much for the lovely, lovely love that you had given me, and please take care of yourself. Please make sure you send this particular video link to all of your other friends who are struggling with principles of inheritance and variation, and we are going to cover it together. I hope you enjoyed the session. You're going to get the PDF in the WhatsApp channel as well as in the AIO page, so make sure you go join both places. Okay, so yes. Yes, you can try explaining your issue. Thank you, ma'am. Today's schedule, you're talking about, so tomorrow I know what class you have. I think you have it for mathematics. Tomorrow you have a math class. Okay, yes, I'm waiting. Tomorrow you would be having a math session. My classes are going to be there on Wednesdays. Okay? Yes, I'm waiting for you. Hello? Illa anige class ഞാൻ ഒരു 5 മിനിറ്റിൽ ഇറങ്ങും. Okay? Okay, bye. Okay, Priya. Bye-bye. See you. And yes, I'm still waiting for you. Yes, Pawan, I'm teaching from Bangalore. Okay, whenever I fill my 10th roll number and proceed ahead, it's not proceeding ahead. Okay, so yes, apparently that should not be happening. Are you filling the correct number out there, and have you tried to like checked, because in 10th mark sheet there are multiple numbers? Can you check any other thing which is just got stuck at the same page? Yes, I really do have to check that issue up, because I do not know. Okay, if I come across a solution to that, I'll definitely let you know, but that should not be stopping you from trying. Please make sure you keep on trying, and also check your mark sheet really well. The roll number is the same thing that you are adding up there. Just check through that. Okay, if I get an understanding of what has to be done, I'll surely try it up. Okay, will that work? Or also, did you check the eligibility criteria before applying? Please do check that as well. Okay. Akash, hello. Yes, ma'am, I'm filling after reviewing. Okay. Yes, if possible, definitely I'll let you know a solution for it. As of now, I have no solution for it. That's why I'm just not talking much about it. Yes. Cool. So in the next session probably, or if you comment down somewhere in the comment sections, I possibly could be replying to you if I find a solution, not giving a guarantee whether I can or not, because I do not have the interface for the NEET thing. But yes, if so many of you are experiencing that, probably they could be telling something about it, and I'll let you know about it. Yeah. Okay, thank you so much. And Akash dear, the class has ended, Macha. Now it's time to say bye. So I'm saying bye, and I'm saying wishing you a good night ahead. Please make sure you sleep tight and do not let the mosquitoes bite. Bye bye. Lots of love. Join the WhatsApp channel for getting updates. Bye bye. Waiting for you guys to join the next session as well. Bye. Yes, Akash, tell me. First. Yes, you can do that. Okay, only if I find the solution, I'll be giving you a reply comment. Okay, if I don't, probably I'll say that I do not know it. Okay, so Akash. Oof, ma'am, I love fluid without FLID. Oh, thank you so much, Da. I love you all too. Okay, bye. Thank you. Bye-bye. See you.